Okay, I sent these emails. Oh, people is arriving. I see that participants are raising. So I will leave the floor to welcome from my side. I'm Marco Falconi, member of the Scientific Committee of Remediate Europe. Welcome, Sabine. Good to see you and all the other people that are present right now. I hope you a wonderful session. We have a number of interesting speeches. So I leave the floor to Natalia, Dei, and Christian for the presentation of the session. First of all, if Natalia may introduce where we are in the program and what is the general program of today.
Yeah, thank you so much, Marco, and welcome to all the participants to this 5th session of another event of Remtech Europe. Good morning, good afternoon, good evening to all of you. It is a pleasure to be here with you today in this session 5 about bioremediation and phytoremediation in agricultural, industrial, and military sites. Today is 21st of September, and we will start with 7 presentations for today. My colleagues, Christian Vermeil and Deji Hu from Switzerland and Tsinghua University will introduce the speakers afterwards, but I just want to brief you on how we will behave in this session. We will have the 7 presentations on a row, and then afterwards we will have time for a couple of questions, but you are invited to post your questions on the chat after each or during each presentation. So the speakers are also invited to address the questions that are on the chat and also be ready for a few questions after all the presentations that we will have. Feel free to raise your hands in case you want to make a comment at the end of the session, and we can give you the floor as well. So participants and speakers, it is a pleasure to be here with you today, and I hope that we will enjoy a very fruitful session. I just want to give the floor to my colleague Christian and Deji for them to introduce themselves. By the way, my name is Natalia Rodriguez. I'm from the Food and Agriculture Organization of the United Nations and part of the Global Soil Partnership. So Christian, welcome and good morning.
Thank you very much, Natalia. Good morning, everybody. It's a real pleasure to moderate this session this morning. And I'm Christian Vermeil. I've been working for the Swiss government at the Federal Office for the Environment, and I'm the head of the section Contaminated Sites. Please, Dei.
Thank you, Natalia and Christiana. Hi, everybody. My name is Dei Hou. I'm working at Tsinghua University in China. Tsinghua is the best university in China, but some of you haven't heard of. I hope you can learn more about our university. And my research has been focusing on soil and groundwater remediation. I'm very happy to have this chance to co-chair this session. I will introduce the first 3 speakers, and then Christiana will introduce the following speakers. First, let's welcome Awalu Hassan from University of Malaya, the Institute of Biological Science and Faculty of Science. And the presentation is entitled Combined Effect of Prosopis Duriflora and Consortia of filamentous fungi on the remediation of metal impacted landfill soil. Owalu, you can share your screen and start your presentation.
Okay, thank you very much, sir. Can you see my slides?
Not yet. Okay, we can see it now.
Okay. Okay. Thank you very much. Good afternoon and good evening, or good morning to all of us. I'm here to present on a topic, combined effects of Prosophis juliflora and consortium of filamentous fungi on the remediation of metal-impacted landfill soil. Sorry, it's very slow. As we are aware that increased industrialization and urbanization have led to increased generation of municipal solid waste, with the current global estimates of 1.3 billion tons per year and is expected to increase to approximately 2.2 billion tons per year by 2025. MSW landfills are the primary repository for disposal of waste worldwide, including in countries such as Malaysia. Now, due to the composition of MSW, during decomposition, leachate is generated. An estimated 13 million liters of leachate is generated every day in Malaysia. Here we can see a typical leachate generation at Tamambringi landfill. This leachate is contaminated by various toxic pollutants. Now, among the contaminants in the leachate, heavy metals are of worrying concern, and these heavy metals are known to cause serious environmental problems. As such, they need to be removed from contaminated land. And this can be achieved using an efficient remediation option. That's phytoremediation. In this case, plant and fungi. The research is therefore aimed to demonstrate the combined effects of Prosophis juliflora and consortium of filamentous fungi on the remediation of metal polluted landfill soil. Now, Tamambringi landfill, which is located at Jingjang, Utara, Kuala Lumpur, was selected for this research. Soil samples were collected from different points within the landfill and transported to the laboratory for analysis. And some of the parameters analyzed in the laboratory were pH conductivity, redox potentials, metal concentration, and microbial population. And the plant used for the experiment was also collected from the Tamambringi landfill. The formulation of the fungal inoculum involved 13 species of identified fungi. These fungi were used to make up the inoculum of consortium. Now, this inoculum was used for the phytoremediation experiment. The phytoremediation setup involved treatment and control. The treatment consisted of 3 kg of contaminated soil, P. juliflora, and 300 ml of fungal inoculum. Meanwhile, the control consisted of 3 kg of contaminated soil and P. juliflora, and there was no fungal amendment. The setup was kept for 120 days, during which monitoring activity was carried out at day 0, 30, 60, 90, and 120. And at the completion stage, some of the parameters analyzed were physicochemical parameters and microbial population. Now, at the completion stage of the experiment, the plant was harvested and analyzed for various parameters, and that's the completion of the experiment. Now, the results. Here we have plant growth parameters. We can see that the shoot and root line of P. juliflora or inoculated P. juliflora were found to be significantly higher than those of the control. And similar result was noted for the shoot fresh and dry weight. Now, meanwhile, in the case of the root fresh and dry weight, we observed that the inoculated P. juliflora or the root of the root fresh weight of inoculated P. juliflora was found to be significantly higher than that of the control. However, with regard to the root dry weight, there was no significant variation between the treatment and control. Now, here we have metal uptake by P. juliflora. We can see that all the metals were substantially accumulated in the inoculated or in the shoot of inoculated P. juliflora as compared to the uninoculated one, which indicated the likely influence of the inoculated fungal organism on the increased accumulation of these metals and subsequent translocation to the above ground part of the plant. Meanwhile, in the case of the root, we can see that the iron was better accumulated in the roots of an inoculated plant as compared to the inoculated one. Meanwhile, in the case of zinc, it was better stored in the root of inoculated pidgin flora as compared to the control. However, in the case of the nickel and lead, there was no significant variation between the treatment and control. Now, here we have metal accumulation characteristics of pidgin flora. Now, based on bioabsorption coefficient, we observed that all the metals were significantly absorbed from the root, from the soil to the plant, by the plant. Meanwhile, with regard to the translocation factor, we can see that all the metals were substantially absorbed and translocated to the above ground part of the plant, which indicated that the plant is better at phytoextraction of this plant. Meanwhile, with regard to the control, in the case of the bioabsorption coefficient, we can see that only lead was substantially absorbed from the soil. Meanwhile, others were less absorbed from the soil. Now, with regard to the translocation factor values, iron was more phytostabilized by the plant. Meanwhile, nickel, lead, and zinc were better phytoextracted by the plant. Now, in case of the residual metal concentration in the soil, the treated or the residual metal concentration in the treated pidgin flora in the treated soil was significantly lower as compared to those in the control, which indicated the likely influence of this inoculated organism in enhancing the absorption of these metals from the soil. Now, here we have the physicochemical and biochemical characteristics of pidgin flora. Now, with regard to the chlorophyll content, we observed that the chlorophyll content in the treated pidgin flora was significantly higher as compared to that of the control. And this may be the result of the reduction of the metal toxicity by the inoculated fungal organism, which enhanced the photosynthetic activity and hence the result of beta growth, as we noted earlier. Now here, this is the carotenoid content. Although the carotenoid content in the control plant was higher than that of the treated plant, however, the difference was not substantial. Meanwhile, with regard to the proline content in the root of inoculated pidgin flora was significantly higher than that of the control. Meanwhile, on the other side, the leaves or the content in the leaves of control was better than that of the treatment. Now, antioxidant enzymes, we can see all the antioxidant enzymes in the treated plant were substantially, or the activities were substantially higher. This shows that these antioxidant enzymes were, the production of these enzymes were stimulated higher in the treated plant, likely as a result of the inoculation of this fungal organism. And this might have reduced drastically the toxic effects of these metals, and thereby enhancing the growth of this organism, of the plant. Here we have other metabolites. We can see here we have the tetrahydroxyflavone and the hydroxyflavone, which are members of flavonoid. Those have strong antioxidant properties against many toxic metals. Then we have glutathione. Glutathione has been regarded as having strong antioxidant property, and in addition, it has very high, or rather high absorption, binding capacity to many metals. We have tryptophan and indole acrylic acid. Tryptophan has been considered as a precursor for the synthesis of IAA in many microorganisms, and this IAA has been considered as an important growth hormone. Meanwhile, indole acrylic acid has also been considered as an important growth hormone. All these might have helped in the reduction of metal toxicity, thereby enhancing the growth of the organism and of the plant, and also the absorption of the metals. Now, we have endoparticle root colonization. The frequency of root colonization, intensity of root colonization, and avascules and vesicles abundances were found to be substantial in the inoculated pidgin flora as compared to the control, which indicated that the inoculation of this fungal organism enhanced the activity, or rather enhance the association between these organisms, thereby enhancing the phytoremediation ability of the plant. Here we can see extension of intracellular mycelial growth within the root cells. Meanwhile, here we have the vesicles abundance, and here we have the vesicles abundance within the root of the plant. Now, in conclusion, enhanced growth was observed in fungi inoculated purulent flora over the uninoculated one, and the stimulated production of photosynthetic pigments, metabolite, and antioxidant enzymes was also noted in inoculated purulent flora. And the inoculated purulent flora showed phytoextraction potential for all the metals. And the residual metal concentration in depleted soil are significantly lower than that of the control. Therefore, endophytic fungi play significant roles in enhancing the tolerance growth and phytoremediation potentials of purulent flora in metal contaminated soil. Thank you very much.
Thank you, Awalu. I think you control the time very well. We may even have time for one question or so. Marco, I mean, because we have saved some time, should we ask questions now instead of going to the panel discussion?
No, we go directly for the next speakers. We have 7 speakers, and remind every discussion
follow the rule then. Okay. Thank you, Marco.
Thank you.
Okay. Thank you, Awalu, again, for saving us some time and a great presentation. Next, let's go to our next speaker, Robert Newton. Robert is a PhD student at the Faculty of Environment in UJEP. His research focuses on the treatment and the utilization of agricultural waste for soil improvement and plant biomass productivity. He's involved in the German Czech called the Msw Value project, which focuses on creating value chains for utilization of Msw fibers from sustainably managed marginal and post-mining areas. Now let's welcome Robert. Robert, please share your screen and give your talk. Great, we can see your screen now.
Thank you very much. Good morning, good afternoon, good evening, and thank you for the introduction. And welcome to my presentation on zero waste approach in Wisconsin species phytotechnology applied to effectively contaminated or marginal lands. It's taking some time. Yes. Okay. So I'll talk about phytotechnology with Wisconsin species, properties, and role of biochar, climate change, and soil health. I'll talk about biochar received from Wisconsin's biomass waste, and testing of Wisconsin's biochar, some experiments we are working on, and then conclusions. So to start, Wisconsin species is among the most promising energy crops for phytoremediation in slightly contaminated or marginal lands, which contributes to the improvement of soil health, prevention of soil erosion, and reduction of emissions of greenhouse gases. Due to the high lignocellulosic content of miscanthus, it can be converted into energy and bioproducts, such as pulp, paper, and fiber. And when miscanthus is converted to bioproducts, agricultural waste, such as contaminated rhizomes, unconditioned biomass, and processing waste, can be converted to biochar, which allows for a zero waste approach when it is then returned to the production process via incorporation into the miscanthus field, as shown here in this figure. So generally, what is biochar? So biochar is a carbon-rich product derived from the pyrolysis of biomass with temperatures between 300 and 700 degrees Celsius. It interacts with trace elements by absorbing their ions on its pore surface and transforming them into hydroxide, carbonate, and phosphate precipitates. So the figure here shows various ways biochar interacts with the ions of trace elements in soil. So biochar and climate change. So biochar is growing in research today a lot due to the role it plays in the mitigation of climate change. It serves as a stable way to store carbon in the ground, leading to withdrawal of carbon dioxide from the atmosphere. And the pyrolysis technology, which makes biochar, allows carbon to be circulated in the environment, shown in this figure here, where about 50% of the carbon will be made into biochar, which is stored in the soil, and the rest, the remaining 50%, can be converted into energy, which when used, then the carbon dioxide can be taken by plants during photosynthesis. So impact of biochar to soil health. Biochar stabilizes trace elements in soil. Thus, when amended in contaminated soils with the cultivation of plants for phytoremediation, it reduces the uptake and bioaccumulation of trace elements by plants. In addition, it provides plants with nutrients such as nitrogen, phosphorus, potassium, and others, which enhances plant growth. Biochar in soil can also help decrease plant stress and prolong vegetation. Impact of miscanthus biochar to soils contaminated by trace elements.
Robert? Oh, okay.
Yeah. Sorry for.
Robert, sorry, can you speak nearer to the microphone? We can so that we can hear you better.
Okay, I'll try my best. Okay, so in this section of the slide, basically we are talking about the use of miscanthus biochar, where to support phytoremediation. And studies that tested miscanthus biochar to support phytoremediation of various plants show that miscanthus biochar does help reduce the uptake of trace elements by plants, and this is increased with the increasing application rates. And this figure here shows different studies who have tested miscanthus biochar. So miscanthus, so biochar made from miscanthus are prepared at different temperatures, various temperatures, and tested in amended in different soils and cultivated in different plants. And for example, looking at the first two references, which is here, they show growing of these plants and amend with 1%, 5%, 10%, weight by weight, miscanthus biochar, and test on different trace elements. And the colors here actually indicates the trace elements, so yellow for zinc, blue for copper, green for cadmium, and red for lead. And here you can see as the percentages of biochar increases, the trace elements are uptaken by the plants actually reduces. However, it's not the case for all the studies, where in some cases here, for example, zinc actually increased by the uptake of this plant, which means that some data actually refutes the conclusion that the higher the application rates, then the better, or the reduced uptake of trace elements by plants. So, and I think there should be more studies. We need more studies to have better conclusions in this case. An impact of miscanthus biochar on plant productivity. So, the figure here shows the increase in plant productivity with the incorporation of miscanthus biochar at different application rates. And the results from different studies indicate that plant productivity increased in parallel with increasing application rates of miscanthus biochar. So you have here a difference, 1%, 5%, 10% weight by weight of miscanthus biochar, and it shows that the percentage of control, that the higher the percentage, the higher the plant productivity. Now this shows, these figures here show impact of biochar to miscanthus grown in oil contaminated soil. And this is from results from an experiment done in our laboratory, which showed the impact of different biochars, the impact different biochars have on the growth of miscanthus when cultivated in oil contaminated soil. So, and the oil contamination was actually diesel. And these, the blue bars are actually the diesel in the, and A is actually least concentration of diesel, E is highest concentration of diesel, and the blue bar is high of the plant without biochar, and it shows that it deteriorates with a high amount of diesel. However, with the inclusion of biochar, this deterioration is prevented. And with here, looking at the impact of different biochars to the plant height at the same diesel concentration, the biochar from the mixture of wood and biohumus impacted positively on the height of miscanthus, while biochar from wastewater and sludge showed a positive impact later in the vegetation period. So this experiment in general showed that biochar helps in increasing the vegetation period and decreasing the plant stress. The types of miscanthus waste which are converted to biochar. So we had rhizomes grown in trace elements, contaminated soil, rhizomes grown in the marginal land, and mixed miscanthus biomass. And this was done at the Technical University of Ostrava. And these are some properties of the biochar received from the different miscanthus waste. And for example, surface area is very important for biochar to absorb trace elements, and the rhizome, the biochar received from rhizomes grown in the marginal land shows a higher surface area. And this biochar received from rhizomes grown in the marginal land was tested in soil contaminated with trace elements, whilst the biochar received from rhizomes grown in the trace elements contaminated soil was tested in an experiment with Spinacia oleracea L, which I'll show in my next slides. So this is the experiment with the miscanthus biochar received from rhizomes grown in the marginal land, where 2 doses of this biochar was amended in soils contaminated with copper and zinc. And as at August 2021, miscanthus growing in the soils amended with 5% biochar have shown better development and bioparameters, that is more shoots. This is the pot of soil contaminated with copper and has 5% biochar, which has more shoots compared to 1.66% biochar and one with no biochar. And the next experiment is where Spinacia oleracea L was a testing plant cultivated for a period of 2 months, May and June, in soil amended with biochar received from the rhizomes grown in the trace elements contaminated soil. And plants cultivated in soil amended with the miscanthus biochar from contaminated rhizomes grew better. So showing some images of the vegetation, different vegetation periods, and evaluation and of soil parameters and bioparameters is currently underway. So conclusions. Biochar is important since it contributes to a circular economy and the mitigation of climate change. The production and usage of miscanthus biochar completes a loop of the miscanthus value chain. When used for bioproduct, since the wastes from the field and processes can be returned to the production site via incorporation in soil to provide sustainable use of the feedstock, reduce costs, and ensure a zero waste approach. So thanks to the Connect Instant Value projects and all partner institutions, especially the Technical University of Ostrava and the BURB Institute in Homotov, Czech Republic, for making this work possible. Thank you very much for your attention.
Thank you, Robert. This is a very interesting talk about the application of biochar for soil remediation, and Robert also controlled time very well. Now let's go to the next speaker, Elisabetta Franchi. Elisabetta graduated in pharmaceutical chemistry and technology. She has been working in ENI for many years and has been involved in various activities relating to biology and microbiology applied to biotechnology. She is currently engaged in projects for the remediation of soils and water contaminated by metals and hydrocarbons using biological approaches. Now let's welcome Elisabetta. Elisabetta, we can see your screen, but we don't see the PPT yet. I think you could share your screen and open the PowerPoint, go full screen.
No, I have to share. I tried. So you can see.
We still cannot see it. Okay. We can see your screen now. Could you open the PowerPoint file?
Okay.
Yes, we can see now.
Okay.
Yeah, it works now.
Right now. So the case study we faced was a break-in attempt in an oil pipeline near an agricultural area in Italy. So based on previous experiments with other hydrocarbon contaminated soils, we have planned to carry out a laboratory scale test to evaluate the biodegradative capacity of the indigenous microbial community with different treatments: natural attenuation, land farming plus bioaugmentation, and phytoremediation trial testing the performance of 3 plant species, corn, lupine, and alfalfa. The results obtained have contributed to the preparation of the operational remediation plan. Okay, here is the map of the area with the indication of the sampling points defined after the spillover event to verify the contamination level. The soil sampling points for this experimentation were chosen in the area most impacted by the spillover and based on previous analysis. The points in the yellow circles were those used for phytoremediation trial, and the point in the red circle, SB, the closest to the breaking point, was used for the bioremediation experiments. About 25 kilograms of soil were collected at 2 deepness, 1 and 2 meters, for each point, and the samples were homogenized and sieved at 5 centimeters directly in the field. On the right of this slide, you can see some images taken during the sampling. I have to say that although about 4 years had passed since the spillover, the olfactory impact of the hydrocarbons, especially at the ridge level, was quite intense. After the sampling, we carried out characterization activities. First, we analyzed the contamination levels of the different soil samples, and as shown in the first table, the values of hydrocarbons greater than C12 were of the order of thousands of ppm. The values of hydrocarbons lower than C12 were not significant and are not reported. The second table shows the microbial count of the soil samples. This analysis is essential to verify the possibility to apply a bioremediation approach, and the numbers of colony-forming units per gram showed a good biodegradation potential. The third table contains the soil texture features, very important parameters since the physical characteristics of the soil, in particular the weight percentages of the granulometric classes, sand, clay, and silt, can influence thermal profile, permeability to air and water, redox potential, and surface reactivity. As I said before, for the bioremediation test, we used the soil sample SP, the one closest to the spillover. The soil sample was previously dried under a hood for 4 days and then sealed up to 2 millimeters. This is the scheme of the experiment: plastic trays containing 1 kilo of soil in duplicate were set up for each treatment: natural attenuation, land farming, and land farming plus bioaugmentation. The treatment conditions for the different samples were as shown. For the natural attenuation, the soil was only moistened weekly to keep the water content around 10–20%. For land farming, a mixing was added to support the activity of aerobic microorganisms. The land farming plus bioaugmentation was treated like land farming with the addition of the inoculum at the start of the test. The preparation of the inoculum is detailed in the next slide. For the enrichment of the oxidizing hydrocarbon bacteria present in the soil, a mineral medium with 5% of diesel as the only carbon source was added to a homogeneous sample of soil with aliquots of each point. The sample, 4 replicas, was incubated at 30 degrees Celsius, and after 4 days of incubation, a second enrichment step was performed by diluting the culture in new flasks and fresh medium. After the third step, the diesel droplets. After the first step, the diesel droplets colonized by bacteria were very evident, as shown in this image. And thus, after several washes to eliminate the diesel added, an aliquot of the cells was used to inoculate the land farming bioaugmentation trays. After this, we performed the isolation of the oxidizing hydrocarbon bacteria from this enrichment. Starting from the enrichment, we have prepared serial dilution and we plated them on agar plates. About 20 strains were then isolated, and the sequences of the 16S fragment have identified bacterial genera classified as oxidizing hydrocarbon bacteria. But as most of them belong to risk level 2, only 2 strains, here highlighted in yellow, were analyzed, here plates, were analyzed for the presence of some of the main plant growth promoting properties for the preparation of the microbial inoculum to apply in phytoremediation trials. The table summarizes the results where it is highlighted that both strains show the ability to produce acetic acid, indole acetic acid, siderophore molecules, and ammonia. Here I show the results of the bioremediation test. The 2 diagrams show the biodegradation trend for the fraction C9C18 and C19C36. After 60 days, the decrease in hydrocarbons is already significant. The percentages measured at the end of the test, that is after 180 days, show quite similar values between the different treatments, even if slightly higher for the land farming and bioaugmentation samples. Considering also the percentages relative to linear and branched hydrocarbons, it is evident, as expected, that the more complex molecules, longer and branched, are more recalcitrant to the enzymatic attack, and their degradation occurs more slowly. In fact, as shown here, with the fraction C9C18, the percentages recorded after 180 days were already reached after 120 days. For the phytoremediation trials, since investigation with plants due to the high biological variability requires several replicates, it was necessary to reduce the number of samples to be tested. Therefore, the 16 soils were pooled, considering similar concentrations. 4 batches were then prepared, B1, B2, B3, B4, with the average concentration shown here. So, microcosmic tests with 5 replicates were set up with 3 plant species, lupine, corn, and alfalfa. The first experiment was the comparative growth of plants on contaminated soils and on a controlled soil from organic farming with very low level of hydrocarbons. Comparing the yield of the vegetables, a reduction is noted for all the plants grown on the 4 soils. Considering the aerial part, the maximum reduction is about 38 for lupine, up to 40 for corn, and 33 for alfalfa. Something happens.
Yes, sorry, Elisabetta. Someone started sharing the screen.
Okay.
Can you please put back your slides?
Okay. Can you see?
Yes.
Okay.
Yes, perfect.
Okay. Going down. Sorry about that. Doesn't work now. Okay. Okay.
Elisabetta, oh,
The second experiment was the comparative growth of plants on the contaminated soils without orange bars and with yellow bars. The evaluation of the two isolated and characterized PGPR strains with respect to control soil green bar. The diagrams show the biomass values, fresh weight, of the plants, and the results clearly indicate that the microbial inoculum was able to overcome the growth problems which have been encountered. There is a significant positive effect of PGPR, and the biomass increases under these laboratory conditions up to 50%, mostly in lupine and alfalfa. The production of biomass is similar to that in the controlled soil, and in some cases even higher. The next test was to evaluate the effect of the plants on the biodegradation of hydrocarbons. After previous experiments carried out at microcosm scale, which provided interesting results, we moved on to the mesocosm scale, foregoing step before the pilot field. And this test confirmed a positive effect of the plants. In the diagram shown, you can see the comparison between the starting soil, blue bars, the non-vegetative soils, orange bars, and the vegetative ones, green bars. The experimentation was carried out in triplicate in a greenhouse, and each mesocosm was prepared with 5 kilograms of contaminated soils. All pots were periodically irrigated according to the needs of the plants, and water was also periodically added to the non-vegetative mesocosm, thus mimicking the natural attenuation. These results indicate that the contribution of plants appear to be significant, highlighting an increase in degradation of 15–18% in vegetative soils compared to non-vegetative ones. The samples from the bioremediation trial were also analyzed from a metagenomic point of view. For the metagenomic analysis, we considered the soil at 30, 90, and 180 days the last time. The first evidence to underline is that in the samples, as expected, we found several species of oxidizing hydrocarbon bacteria belonging to the listed orders. As a detail, we can highlight that in all samples, after 90 days, we see a rise of the order Acidomicrobiales. Then, always after 90 days, but only in the treated samples, we can see an increase of the order Sphingobacteriales. At last, in all samples, but especially in R, L, and LB, treated samples, at 90 and 180 days, is evident a rise of the order Actinomycetales. So we come to the conclusions. As for the bioremediation test, after 180 days, we reached percentages between 83 and 96 for linear hydrocarbons, and between 76 and 83 for the branched ones. With the phytoremediation approach, we detected a decrease in hydrocarbons of 15–18%, favored by the vasodegradation activity. And the addition of PGPR inoculum promoted the growth of the tested plants with increase of the biomass up to 50%. The results obtained with this experimentation contributed to the preparation of the operational reclamation plan, in which biological and chemical approaches will be integrated to achieve the best requalification of the area with the restoration of the agricultural activity. Finally, I would like to thank all my colleagues who contributed to the project. And here are some of our recent papers, my email, and thank you to all.
Thank you, Elisabetta. This is a very nice presentation. Next, I'd like to ask my colleague, Christiana, to introduce the remaining speakers. Christiana, please.
Thank you very much, Day. So I have now the pleasure to introduce the next speaker, who is Jelena Avdalovic. Jelena is an associate research professor at the Institute of Chemistry, Technology and Metallurgy at the National Institute of the University of Belgrade. She graduated in biochemical engineering and biotechnology and holds a master's degree on science in chemistry and a PhD on biochemistry. Jelena will now present us a project of bioleaching of metals from printed PC motherboard by Acidobacillus. Please, Jelena.
Okay, thank you. Can you hear me?
Yes, very good. Thank you.
Okay, now I will share my screen. This one. Second to put that on the full screen, but okay. Well,
it's okay. Thank you.
Yeah. Hello once again from rainy Belgrade this morning. I have this opportunity to represent this work. First of all, I have to say that I'm not the first author. I'm the Jelena, but I'm not the Jelena Avdalovic. Unfortunately, she's not available today to represent this work, so I will take the role of the speaker. I'm the fifth author of this small article. My name is Jelena Milic, but I'm the closest colleague of the first author, and we were working together on some parts of this experiment. However, I put here her email address, so because I'm not so sure that I could answer on all questions. For some questions, if I don't have the answer, I will put the email as well in the chat. So we already agreed with Jelena that she will answer on the email. You can just put the subject RemTech conference, and she will know that it is a question from this presentation. So let's get started. This research, this is one initial small research. We just try to do some experiments with bioleaching of rare metals from IT equipment. I will use this opportunity just to present our small team. We are the small group at the Institute of Chemistry, Technology, Metallurgy, which is National Institute of University of Belgrade in Serbia. We are based in Serbia. We are mostly working in the field of environmental protection, and we are working a lot with bioremediation. Actually, we are working with microbes from the environment, so we are checking their ability and how to use their metabolism and ability for sustainable development. So at this work, we focused at Acidobacillus peroxidans. Actually, we focused to can we somehow leach the rare metals from electronic waste. We put here the three main bullet points. First of all, that electronic waste is the fastest growing waste category. I think that all of us are aware of that, because we know that I think that every year we are buying some IT equipment. You need a phone, or you need a tablet, or you need your PC. So how we are buying on that way we are producing the waste as well. Recycling of electronic waste. So here in Europe and in Serbia, Serbia is not the EU, but we have the same laws as EU concerning the waste management. So electronic waste is hazardous waste here because of a lot of toxic chemicals inside of this kind of waste. For example, POPs chemicals, flame retardants, and etc. But however, in this kind of material, we can find very valuable metals, rare metals in Europe. So we don't have sources, natural sources of these metals in Europe, and it is very important for us to find a way to recycle that valuable material from this waste. So we check the use of microorganisms for bioleaching is not something new, but we wanted to try work with Acetobacillus phytoxidans, which is isolated strain from Serbia territory, and we know that this strain is very potent strain for some other things to say, but we wanted to check activity on the PC board for actually IT equipment. So as I mentioned, the aim of this study was to investigate the potential of using the Acetobacillus phytoxidans to solubilize metals from electronic waste. So we had as electronic waste, we took the PC motherboard, and first of all, we need to separate the plastic parts, and the metal parts our colleague shredded. So we have small parts of metals at the end from this PC board. The pre-treatment of this material was in sulfuric acid for 48 hours, and after that neutralization, it is washed with water and dried. As well, we were using pyrite concentrate, because this pyrite concentrate is the natural substrate for this bacteria. The concentrate, pyrite concentrate is from Bor. It is one city in Serbia where we have a lot of mining. So from the border we have pyrite, and the Acetobacillus is isolated in the border area as well. So it is their natural substrate, and we use it as well. And the preparation is very similar as for metal parts of motherboard. So it was treated with sulfuric acid, mixed, and after that, after one night, it was washed out with water and dried to constant mass. And both e-waste and pyrite was used in this experiment, the leaching experiment, which was carried out with Acetobacillus phytoxidans in the period of 14 days, on temperature of 28 degrees and with rotation. Here is some preliminary our result. At the end, we detected the metal in the solution. The most abundant metal was nickel. And I have to say that this small experiment from 14 days shows we can say that this is effective metal leaching. But I have to say that since we were using the Pirate, Pirate has mostly the iron. The concentrate is Pirate concentrate is actually the iron. We don't have these kind of metals in Pirate, so we can assume that all leaching metals from this experiment originates from the PC motherboard. So it is a preliminary experiment just to give us the direction where we can go with this strain which we already isolated a few years ago. Our first conclusion is that this strain of phytoxidants is able to grow in the presence of electronic waste. For motherboard, we can get the copper, nickel, zinc, tin, and cobalt. It can be recovered. But however, for IT equipment, you have a lot of different kind of metals and substrates to use. Maybe some other metal parts from IT equipment have another metals, not the same as motherboard. So in general, investigation of electronic waste bioleaching will be continued in our laboratory in future. But this time we are planning to work with a larger number of microorganisms, because it's going to be Acetobacillus phytoxidans as well, but we have around 10 strains of this bacteria, and we want to check them all. We need to put a longer time of experiment as to have a longer duration for leaching. So thank you for attention, and once again I will put the email in the chat, so you can if there is some question that I don't have the answer at the end of the session, you can send it to the first author. Thank you.
Thank you very much for the presentation of your first results on the project on electronic waste. So there will be probably questions during at the end of this session. We go on now with the presentation. The next is a presentation of Neda Amanat. Neda Amanat is a chemical engineer working as a postdoctoral researcher at the Sapienza University of Rome. She is involved in novel methods and developing engineered solutions for the remediation of contaminated sites. The title of the presentation of Neda is Characterization of Synthetic and Natural Surfactants for Mobilization of Contaminants in Environmental Matrices. Please, Neda.
Thank you. Hello, everyone. I'm going to present our project on characterization of synthetic and natural surfactants for mobilization of contaminants in environmental matrices. This project is a cooperation between Sapienza University of Rome and chemical company that produce and formalize chemical compounds. In general, we can say that surfactant is an amphilic molecule with a hydrophilic head group and a hydrophobic tail. By adding surfactant to the solution, we can observe the reduction of interfacial tension between immiscible fluids, and as a result, we have mobilization of organic contaminants. At the critical micellar concentration that the monomer of surfactant forms micelles, and above of this critical micellar concentration, the solubility of the hydrophobic compounds improve. And the mechanism is when we have a hydrophobic contaminants adsorbed on soil. By adding a surfactant to the solution, we observe absorption of surfactant molecules at the interface between soil, water, and contaminants complexation. Then we will have a desorption of the surfactant contaminant complex and the formation of micelles. And at the end, we have the precipitation of surfactant out of the complex. And as the poor solubility of the organic contaminants is a limiting factor for effective remediation, and also surfactant has been shown to be an effective solution. In this context, surfactant enhanced aquifer remediation has been proposed as a means of enhancing removal of non-aqueous phase liquids by applying surfactant in the in situ flushing. The technology has included injection of surfactant solution to the contaminated site, and then we have as a result, we have the mobilization and solubilization of contaminants. Subsequently, the contaminants and the surfactant solution will be extracted from extraction gas. Then we have the treatment of the surfactant, and we can reuse it in the circle, in the system. And as the surfactant selection is a critical point, our scope of the project is to developing a behavior characterization protocol that could be applied for surfactant selection. The study is identification and characterization of a possible surfactant for in situ flushing. And the project divided in two phases. In the first phase, we started with the characterization of critical micellar concentration study of different surfactants. And in the second part, we evaluated the mobilization capacity of our surfactant, different surfactant in batch and column study. In this case, we select 5 different commercial, non-ionic and biodegradable surfactant prepared by Chemic company by the name of NTO 81, 82, 83, 84, and 85. Coming from different families of surfactant. 82 and 81 are synthetic surfactant coming from alkyl glycosides family. 83 is and 84 and 85 are biosurfactant, which 83 is a sulforolipid, and 84 and 85 are rhamnolipids. At the beginning, we started to characterize our selected surfactant by determination of their critical micellar concentration in a series of surface tension measurements. And from the result, we can see that except for the NTO 81, the other surfactant had a very low critical micellar concentration, especially in the case of synthetic NTO 82 and biosurfactant NTO 84. Then we go through the mobilization study by evaluating the mobilization capacity of different surfactant by assessment of the reduction of absorption capacity on a selected reference adsorbent material in the presence of surfactant. We use the different type of contaminants. Once we use toluene coming from Bitex family, and once we use trichloroethene coming from chlorinated solvent. And the reference sorbent material that we use was pinewood biochar as an alternative to the activated carbon. This material has really high capacity to adsorb that we use it in our laboratory for different studies. At the beginning, we conduct isothermal of toluene on water on pinewood biochar in absence of surfactant as a reference test. Then we repeat the test, this time by adding a surfactant to the solution. Here we can see the result from adding NTO 82 synthetic surfactant by using a concentration below its CMC concentration, and by adding the surfactant to the solution, we can see the reduction of adsorption capacity. We again repeat the test. In this time, we use a concentration higher than the CMC, 5 times of the CMC, and once we use also the 5% of the volume of the surfactant solution, which with NTO82, it was 90 times of a CMC, and we can see the effectiveness of our surfactant to reduce the absorption capacity in the presence of the surfactant in the solution. We repeat the test by using all 5 surfactants, and except for the NTO84, we observe that the excellent ability of our surfactant to reduce absorption capacity, especially in the case of NTO82 and NTO85, we observe higher efficiency of this surfactant, especially at the concentration of 5 times of their CMC. And then we go through the experiments and use another type of contaminants, perchloroethylene. Here we observe the higher effectiveness of our surfactant, as we can see the significant reduction of absorption capacity in the case that we use PCE. Also here, the surfactant 82 and 85 showed higher efficiency to reduce the absorption capacity. Then we go to the continuous mode to evaluate the mobilization capacity of our selected surfactants to mobilize the adsorbed contaminants, and also to simulate the soil flushing technology. In this part of the study, we started by the contamination phase of the columns that we filled with sand and interest percentage of pine wood biochar to have a maximum adsorption condition, much more higher than in the soil. And we use different type of contaminants. In the second phase, we change the feeding solution with the flushing solution to have a desorption and flushing phase. Here you can see the result from the contamination phase of the column that contaminated with toluene. This is the breakthrough curve, and when we arrive to the equilibrium where the effluent concentration was equal to the influent concentration by integration this area, we calculated the milligram of toluene adsorbed on the column on the biochar. After the equilibrium, we changed the feeding solution with the surfactant solution that here we use the NTO82 at 5 times of CMC, as we observed that at this concentration our surfactant was effective. And remarkably, after changing this feeding solution, we can see the significant increase in the concentration after a couple of hours, and this shows the high effectiveness of our surfactant to mobilize the contaminants. Also here, by integrating this area, we calculate the milligram of contaminants that mobilize. We repeat the test where in the presence of different contaminants, once toluene and the other times perchloroethylene, and by using different type of surfactants, once we use NTO82 and the other times we use NTO85 coming from biosurfactants. As a reference, also we use water for the flushing and as a flushing solution for having the reference. We can see in general the desorption phase with the surfactant always took place with increasing in the concentration, and the desorption phase desorption phase in the presence of surfactants always was shorter than the contamination phase, and also it was shorter to the desorption phase in the presence by using the water. We also observed that the removal capacity is much higher in the presence of surfactant, especially in the case that we had the PC as a contaminant. So at the end, we can say that our surfactant had a very low critical micellar concentration, which is very important in the economical point of view. We observed that our surfactant had a significant capacity to mobilize the hydrophobic contaminants, and they show the ability to reduce the interfacial tension between non-aqueous phase liquid and aqueous phase. And in general, the synthetic surfactant NTO82 and biosurfactant NTO85 showed the distinguished results and had much better mobilization capacity. In conclusion, we can say that the biosurfactant NTO85, which showed greater ability to mobilize and dissolve contaminants and had higher efficiency of removal of contaminants. In the case of toluene, we had 92% of removal, and in the case of PC, we had 100% percentage of removal. And as this material has a lower environmental impact, which doesn't absorb on soil colloids, and as a result, we have the reduction of secondary contamination. We can possibly consider it for the use in SER technology. Thank you very much for your attention.
Thank you very much, Neda, for your interesting and clear presentation on the use of surfactant. I have now the pleasure to introduce the next speaker, who is Neil Bruce. Neil Bruce is currently Professor of Biotechnology and Director of the Center for Novel Agricultural Products at the University of York in the United Kingdom. The major research themes of Neil's laboratory are environmental and industrial biotechnology. The title of the presentation of Neil is Diffusing and an Explosive Issue, Engineering GM Plants to Contain Explosive Pollution on Military Ranch. Please, Neil.
Thank you very much for the kind introduction, and good morning, everyone. I'm presenting today on behalf of CERDIP and ESTCP, and I'll just first of all like to introduce our project team to you. So there's myself and my colleague Liz Rylett at the University of York, Stuart Strand and Long Zhang at the University of Washington in the US, and Tim Carey from Erdit Krell of the US Army Corps of Engineers. So I'll just give you a brief outline of my presentation to start with. So
Neil, could you put your presentation on presentation or full screen mode, please?
Ah, okay, sorry.
No problem. Perhaps you have to close the share of your screen and then to open it again.
Yeah, I'll do that. Is that better?
It's better. Thank you very much.
Thank you. Apologies for that. Yes, so I'll just give you a brief outline of the presentation. So I'll just touch on the problem of explosives pollution, and then tell you about some of the work we've been doing on discovering enzymes that degrade explosives from our understanding of plant and microbial metabolism of explosives. Then I'll tell you about how we've been using that fundamental knowledge that we've gained from these metabolic studies to engineer transgenic plants that have the capability of remediating explosives on military ranges. And then I'll tell you about the results that we've got from our field trials with these transgenic plants at a military site in New York State in the United States. So first of all, the problem. Explosives like RDX and TNT are highly toxic, and they are recalcitrant to degradation. So therefore they persist in the environment for long periods of time. And as a result of their use over the 100 or so years that they've been around, there's now large-scale contamination of land and groundwater around the world. And a lot of this pollution has been historic from the manufacture and storage of explosives. But what I'm really going to focus on today is contamination and pollution of military ranges. So when a bomb or a mortar round explodes, you never get complete detonation of all the explosives. So you get particles of explosives scattered around on the ground. And when it rains, these particles dissolve, they contaminate the soil, and they can contaminate the groundwater. And this contamination of groundwater can then go on to impact on drinking water supplies, as was seen at the Massachusetts military reservation in Cape Cod in the United States. So there's a real need to develop low-cost sustainable technologies that can be used to remediate these sites, or importantly, to contain these pollutants so they don't migrate off the site. This is an intractable problem because the military obviously need to train with live munitions, and so you've got continual input of pollutants into the environment. So plants have a number of attributes that in some ways make them ideal for this application. You can grow them over large areas of land. They require sort of minimal inputs, and they can be used in situ. And of course, they're a relatively low cost of a low cost sort of means of remediation. However, there are a couple of major challenges for using plants to remediate explosives pollution, and one is their toxicity. So in particular, TNT is highly toxic to plants. So what you can see here is soil contaminated with 100 milligrams of TNT per kilogram of soil. And you can see that these plants, which are Arabidopsis plants, are severely stunted. And this is what they would look like if they were grown in soil that wasn't contaminated with TNT. But in fact, this soil does contain TNT, and this is a mutant line of the plants that we identified that shows enhanced resistance to TNT toxicity. And we mapped the mutation to a gene that encodes for an enzyme called monodehydroascorbate reductase. And this enzyme is plant-specific, and it's found in the mitochondria of plants. And its innate role is involved in ascorbic acid recycling. But when TNT is present, it converts TNT to a nitroradical, a TNT radical, which then reacts with molecular oxygen to create reactive oxygen species. And in doing so, that TNT radical is converted back to TNT, and then this goes round this cycle again. So you get this futile cycle of causing generation of reactive oxygen species, which are highly damaging to cells. And so these therefore destroy the mitochondria and ultimately will kill the plant. And so the TNT goes up into the roots. And so you can see here with the wild type plants, the roots in the first instance are severely stunted before the plant then dies. So these mutant lines show enhanced resistance to TNT. Plants do have an innate resistance to TNT toxicity. They can detoxify low concentrations of TNT. And we've been elucidating the pathways by which they do this. And the major detoxification pathway for TNT in plants is, first of all, by an enzyme that acts as a nitroreductase to form these derivatives of TNT. And that activity adds functionality to the TNT molecule so that the plant can then add glucose molecules. And these glycoconjugates then get bound up in the plant cell walls, so they're no longer toxic to the plant. So that enables us to develop a strategy by which we could engineer these plants so that they were able to take up and detoxify high concentrations of TNT. So what you can see here in this picture, here you have the wild type plants growing in liquid media. And this is our genetically modified line, our transgenic line, expressing a nitroreductase. In fact, it's a bacterial nitroreductase. There's no TNT present in this media. So in the presence of 250 micromolar TNT, you can see that the wild type plants are now dead. They haven't really removed any of the TNT from that medium, whereas the transgenic plants, they're green, they're healthy, they've increased in biomass, and importantly, they've removed all the TNT from the medium. And they will do this at pretty much saturating concentrations of TNT. So this worked remarkably well. So then we went on to develop transgenic plants to degrade RDX. The challenge here is that plants have very limited or no activity towards RDX. So our strategy here was to take microbial enzymes that have activity that degrade RDX and express them in plants. And in a prior project, we'd identified bacteria from RDX contaminated sites that were able to grow on RDX as a sole nitrogen source for growth. And we identified an unusual enzyme system that degrades RDX. And we called this enzyme XPLA. And it's an unusual enzyme because it's a cytochrome P450, but it doesn't use molecular oxygen to degrade RDX. And what this enzyme does, it reductively clips off two nitro groups off the RDX molecule, and it forms this unstable intermediate that basically just falls apart to produce these metabolites, N-diazolbutanol, NDAB, nitrite, and formaldehyde. And the plant is able to metabolize these metabolites and effectively mineralize RDX. So this figure here just shows, summarizes our strategy to engineer transgenic plants for the remediation of TNT and RDX. So we're expressing a bacterial nitroreductase that enables the TNT molecule then to be conjugated to sugar and locked up in the root biomass. And then we express these RDX degrading enzymes, XPLA and its partner reductase XPLB, which enables RDX to be taken up and broken down to non-toxic metabolites. So these are Arabidopsis plants, and what we have here is wild type plants. The plants in this flask have the genes for RDX degradation. The plants in this flask have the genes that encode for enzymes that detoxify TNT, and the plants in this flask have all the enzymes required for detoxification of TNT and the degradation of RDX. And the medium contains TNT and RDX. And you can see here that the wild type plants are dead because of the TNT toxicity, and they've removed very little TNT from the liquid medium. The plants in this flask contain XPLA and XPLB that degrade RDX, but the plants are dead, again, because of TNT that's present. And so no TNT, or very little TNT is removed, and no RDX is degraded. With the nitroreductase lines, these are able to detoxify the TNT. You can see that they're green and healthy, and within 2 days, they've removed all the TNT from the liquid medium. And with the multiple transgenic lines that are expressing all the enzymes, you can see that these plants have removed, again, the TNT within 2 days, and then they go on to remove all the RDX from the liquid medium. This work here is just an experiment where we're doing effectively the same things, but in soil, and the results mirrored what we see in our hydroponic liquid culture systems. So this is a really exciting and important result, because we could show that we could engineer plants to remove both types of explosives pollutants. So the next experiment we did, which was quite a simple experiment, but was an important experiment in the context of our project. So what we've done here is effectively trying to mirror what would happen on a military range. So we've got soil that we then contaminate with RDX. So we water in RDX, and we have our wild type plants, and in this case, just our RDX degrading plants. And then we leave the RDX in the soil for 7 days, and then we water it through and measure the RDX in the leachate. And here you can see that when we flush the RDX through in the non-plant control, you can detect the RDX in the leachate. The same is true for the wild type plants, but with our best transgenic lines, you can see that there's very little RDX being flushed through the soil being found in the leachate. This is because the plants are taking up the RDX and degrading it. So that was, again, a very important result. Now, Arabidopsis plants it's a model plant system for laboratory studies, not appropriate for remediation in the environment. So we decided to express our explosives transforming genes and degrading genes into switchgrass. The reason why we wanted to use switchgrass is that these plants are found, they're native to many US military ranges. They can grow on marginal land, they produce large amounts of biomass, and they produce these dense root systems that cover a large surface area that will grow to quite a significant depth. So we've expressed here our RDX degrading enzymes, XPLA and XPLB, and these are liquid culture studies with our transformed switchgrass. And you can see that the results really match what we found with Arabidopsis very nicely. So the wild type plants will take up some RDX, but not very much, whereas the transgenic lines take up RDX and completely degrade it. So with the wild type plants that are taking up RDX, the RDX just goes up into the aerial parts of the plant where it just accumulates to a certain extent, but it's not degraded. So we can detect RDX in the leaf tissue of wild type switchgrass, but we don't detect any RDX in the transgenic grass. We then did soil studies with the switchgrass, transgenic switchgrass, and again these mirrored our results with Arabidopsis really very nicely. So this is where we've watered in RDX and then we flush it through after a period of time. And you can see that with the wild type plants, we are seeing RDX in the leachate, and we find RDX in the tissue, whereas with the transgenic lines, we're seeing no detectable RDX in the leachate and no detectable RDX in the leaf tissue. So this is well and good. We've shown that proof of principle here, but in order to demonstrate the efficacy of these plants, we needed to do field trials and test the plants in the field. And we were able to carry out these trials at a military site in New York State. We got a permit from APHIS of the US Department of Agriculture to carry out these trials with transgenic plants. And what you can see here is an aerial view of the plots. There are 27 plots. These plots are 3 meters by 3 meters. They're lined to prevent contamination leaving these plots. And what you can see is that we've got a number of lysimeters in the soil so that we can take water samples to measure the RDX concentration. We've got a culvert here that contains a pump so that if we get heavy rainfall, we can pump water into a holding tank so that we don't waterlog the plots. And likewise, if the plots need watering, we can irrigate the plots from the water that's being contained in the holding barrel. We added the RDX by mixing it with sand and spreading it on the surface to mimic what we would find on a military training range. So what you can see here are the plants that went in the ground in late summer of 2016. And they took until about 2018 to get fully developed. So we started taking samples in the growing season of 2018. And what you can see here are our plots, representative images of our plots. So here you have the unmodified, the wild type switchgrass growing in a plot with no RDX. And here's the transgenic lines growing in a plot with no RDX. And here you have the wild type growing in a plot with RDX present. So this is 100 parts per million of RDX, which is about the sort of concentration of RDX you'll find around a low order detonation of munitions on a training range. And here's the transgenic grasses growing on the RDX contaminated plots. So we took water samples, regular water samples from the soil, and we took leaf tissue over the growing season of 2018. We've accumulated a lot of data from these plots, which obviously there isn't time to present today, but if you want to look at the data and find out more about the study, then the work was recently published earlier this year. But I'll just give you some highlights. So our results really mirrored our glasshouse studies, our greenhouse studies really very nicely. So we found that the wild type plants in the RDX contaminated plots accumulated RDX in their biomass as we expected, whereas the transgenic lines, we found no detectable RDX in the leaf tissue because it's taken up and degraded. We also calculated the amount of RDX that was removed from plots. And we can do this because we know the amount of water that's transpired by the plants over the growing season. We know the concentration of RDX in the soil water from the samples that we've taken from the lysimeters. So the transgenic plants remove 6 times more RDX from the plots than the wild type plants. And this equates to approximately 27 kilograms of RDX being removed per hectare of soil. I think this is really very impressive considering this is an unoptimized system. And we know from our previous studies that whilst the wild type plants are accumulating RDX, they're not being significantly degraded. So in the field, these plants would naturally die and the RDX would be returned back to the soil. The other important point about this accumulation of RDX as well is that on a military range, all the plants there will be accumulating certain levels of RDX, and these will then be made available to herbivores that will be feeding on these plants. Whereas obviously with the transgenic plant lines, there's no RDX present in the leaf tissue, so herbivores won't be exposed to it. So in summary, we've developed transgenic plants that can remove RDX and TNT from soil, preventing their leaching to groundwater. We've demonstrated this technology in greenhouse studies, and we've successfully translated this technology, specifically in this case for RDX, at a military site in the United States. So you might be asking, what's next? So now we're gathering data that will allow us to apply for a petition to APHIS to get a permit to allow us to use these plants on a much larger scale. So thank you very much for your attention, and I look forward to answering any questions in the question and answer session.
Thank you very much, Neil, for this very interesting presentation. And I have now the pleasure to introduce our last speaker for the session of this morning. I would like to introduce Msw Vakari, who is Associate Professor of Sanitary Environmental Engineering Department of Civil Engineering, Architecture, Territory, Environmental and Mathematics at the University of Brescia. The title of the presentation of Msw Vakari is a treatment of petroleum contaminated soils using novel rhamnol pills produced on winery residues, a green approach within the circular economy framework in Italy. Please, Msw. We cannot hear you yet.
And now?
It's good, thank you.
Good morning. Yes, thank you very much for this nice presentation, but this is actually for my professor, Msw Vakari. My name is Elif Shebi. I'm a postdoc researcher, actually based at University of Brescia now, at the department Decatam. So I'm so happy today and delighted to give you this as part of our project CREAMO, entitled Treatment of Petroleum Contaminated Soils using novel rhamnolipids produced in winery residues, a green approach within the circular economy framework in Italy. So just to let you know that this project is designed to be a collaboration between two universities in the north, the University of Brescia and University of Milano Bicocca and Enia in the south of Italy. Actually, I'm sorry for the sound. Okay, hopefully it can stop. Okay, so I will continue. So
we can hear you. Thank you.
Okay, thank you very much. So this project, actually, we are aiming to use non-pathogenic bacteria to produce rhamnolipids from wastes coming from the agriculture sector, especially from two potential sectors, that olive oil and wine residues. And the output of our project, we are aiming to enhance, I would say, the bioremediation and remediation processes in the soil. And this is in this project, we are trying to involve all the SMEs and stakeholders acting in this field to increase the awareness about the circular economy, and we need to find a link between this two area. In this project, you know better than me that here in North Italy, we are leading. This is the production of wine, and countries like Italy and Greece and Tunisia, they are, and some Mediterranean countries in general, they are leading. This is the production of wine and olive oil. Here, if you want to implement this is principle of circular economy, you will not find a better place in this. And like a joke, I will tell you, I was so happy to discuss with stakeholders acting in the wine production. They tell me that the people they consume wine here in Italy, they are so educated, and they need to see this is label of circular economy when they consume this product. And this is why we are acting in this field. Just to give you some numbers about the huge amount of wine that it's produced every year. We are at around more than 1.5 million hectoliters in 2019. And here, only in Lombardia, we are more than 21,000 hectares of wine production. And you will have 2 tons of grape marks per season produced. So imagine the huge number of wastes that you will have every season from the production of wine. And in general, I would say to present for you some, I would say, introduction about biosurfactants, but I'm so happy to be after Nada Amanat. She presented this area. But just to tell you about biosurfactants in general, there are several classes like glycolipids, lipopeptides, phospholipids, and fatty acids, and there is other polymeric bacteria. And all of them, they share this as a particular structure, the hydrophobic tail and the hydrophobic head. And when they reach a critical micelle concentration, they could form this particular structure. This is the micelles. But at the moment, we are more or less crippled at the step of production and characterization. We don't see a lot, I would say, large scale application of biosurfactants in the field of bioremediation. And also, there is not a real study of benchmarking of biosurfactants. And this is one of the aim of our project, actually, to go with glycolipids because they have lower toxicity profile in comparison to other classes of biosurfactants, and they are not so harmful for bacteria and microbial communities inside the site. So just a brief introduction about rhamnolipids. You know, most of you that there is dirhamnolipids and monorhamnolipids, and the famous producer here is Pseudomonas aeruginosa. And after production and extraction of this biosurfactants, they confirmed a lot of hotspot of applications from bioremediation to pharmaceutical, therapeutical, detergent cleaners, going even to agriculture. Maybe 5 years ago, I was using Pseudomonas aeruginosa, but you know better than me that if you go to apply broths with Pseudomonas aeruginosa, you will have problem. You need only to extract biosurfactant, which is safe, and apply it. And it could be a costly process. Just to give you some, to inspire you about the field of biosurfactants and the market, there is a huge need now to go for some bio-based products and to apply them into the field. I will not make advertisement for some biosurfactants in the market, but they could compete even with chemical surfactants and regarding their prices. I will not repeat the same thing said by Nade, but just to let you know that this biosurfactant, they had good ability to emulsify hydrocarbons and also to help the biodegradation of hydrocarbons into the field by accession to bacterial cells. As I told you, more than 90% of data now you will see in the field of paramnolipids that are coming from Pseudomonas aeruginosa, and there is other producers. I don't know why they are not well studied, and there is a lot of application of this field. So in this project, we try to shed light in some safe producer like Burkholderia and Paraburkholderia and others, and we try to characterize them and to use them to make like screening steps and to choose one best producer. Before going to this, you know, when you use agriculture waste, you need to be sure that you know exactly the component that you have inside this is matrix. And we showed here in collaboration with Mac Institute here in Italy that non-fermented grape mark, they're rich in glucose and fructose, and they have low alcoholic grade, and they could be used also this is good. They could be used as substrates by bacteria to produce biosurfactants. So we make like screening with the strain named Burkholderia thalensis, which is a safe producer of rhamnolipids. And we made several steps of screening, and we showed that it's able to produce surface tension using sugar as soil carbon and energy source and to produce biosurfactants. And on our substrates, we showed here, as you see in these graphs, that is able to produce rhamnolipids by reducing the surface tension up to 35 millinewton per meter. And we have a good emulsification capacities. Even for the olive pomace, we have also promising results, as you see here, using this strain, and which is the safe strain. I repeat all the time about this. And even the yield of production is good using the first substrate, which is the non-fermented grape mark, as you see here in this graph. And even the purity, it's very good after the purification, which is good. I would say open new ways of application of this biosurfactant. The stability is also in comparison to chemical surfactant, to commercial rhamnolipids in the market, it's very good against pH, salinity, and temperature. And we characterize it also the congenerians that we have of this rhamnolipids using mass spectroscopy, and we found we detected them, because when you use agriculture waste, you need to be careful and you need to well detect your biosurfactants inside the broths. So as you see in this table, we added another I would say knowledge that Burkholderia is good candidate to produce biosurfactants using grape marks as soil carbon and energy source, and the yield it's quite good. So now we are coming to the most the application, and we have several windows. We have going from soil washing into other biological biotreatment like mesocosm biopeel or bioslurry experiment, and even to enhance the bioelectrical systems. So just to give you some preliminary results regarding the soil washing, as you see here, we made like experimental design without biosurfactant, with one first concentration and second concentration more higher, and we found promising results, as you see here. After incubation for 30 minutes, we have a good remobilization using this as biosurfactants. I remind you all the time that producing on winery residues, and using this as to higher concentration. And even for the first one, it's a real contaminated soil here from Italy at around 6,000 ppm, and another one it's contaminated with polyaromatic hydrocarbons, and we have also a good remobilization. So just to let you know that when you apply this as biomolecule, you will have other advantages, that you will find a solution, I would say, for effluents coming from this process, because you will enhance the biodegradation of this effluents after the treatment. For sure, when you have bio-based compound, not only surfactants or other products. So just to give you that in literature recently, a lot of studies they showed when you use supernatant coming from Pseudomonas aeruginosa, you will have a good remobilization also for hydrocarbon. And this is here we showed even with Burkholderia, which is another class of it's rhamnolipids, but they are long chain rhamnolipids. We have the same, I would say, results, and this is encouraging because we can apply directly broths coming after the fermentation on winery without needing to extract. Just to let you know that we have also other activities now are in process because we succeeded to go for larger scale and the production for this rhamnolipids on winery residues, and we are applying them now, and we are seeing the studying them by molecular studies to see the microbial community structure during the degradation of pollutant, and we will see the hydrocarbon content, and I'm eager to see the results. Okay, stay tuned. You will see. I hopefully to have good results in the future. And also we are now going back to see, because recently there is increased interest to study the to enhance the bioelectrical systems activities by use of surfactants, biosurfactants actually, and this is rhamnolipid. They could be good candidates in this field, and this is long chain rhamnolipids not studied before. So we are now at University of Brescia. We are investigating this field, and hopefully it could be a promising subject. So just to summarize, that non-fermented grape marks and olive oil would be good substrates for the production of biosurfactants. Those biosurfactants coming from this safe producer, they could be good. We can apply them into the field because they are so stable, and they have good similarity with the biosurfactants in the market coming from Pseudomonas aeruginosa. And also we are on the way to replace biosurfactants from Pseudomonas aeruginosa. And I would say, as I said earlier, that the soil washing application indicate a good remobilization capacities, like said by Nade, and this would help to improve the biodegradability of soil washing effluents in the future. If you are interested in this field, we published 2 recent papers to summarize those results. I would say this is part of project. It's collaboration between several researchers in this field. I would name Professor Andrea Franzetti, Professor Menturi Vaccari, Franco, and other groups from ENIA, Dr. Silvia Sbafoni. And I would like to thank also the Foundation Cariplo for funding us and inspiring us in this field, and they helped us to have more idea in the future to act in this is circular economy, and this is hot field. And thank you very much again for giving me this chance to be with you, and I'm eager to see your comments. Thank you very much.
Thank you very much, Ali, for this very interesting presentation, and I'm sorry for the incorrect introduction. Now I let the floor to Natalia to open the panel discussion.
Thank you so much, Christian, and thank you to all the speakers. It has been a really interesting session, and I'm sure that many of the participants will have questions to post. There were some on the chat that I would like to raise. For those working on surfactants, some participants ask if you have considered the toxicity of those surfactants and the possible impact even for biosurfactants in soil properties. So I think maybe we can start with Ali, and also, sorry, I cannot see the agenda. Okay. Yeah, maybe Ali, you can start responding, and then some other colleagues can intervene as well for this question.
Yes, thank you very much. Just to let you know about rhamnolipids, they showed their lower antimicrobial activity in comparison to other surfactants, and this is why we are intending to apply them into the field. And this is one of the, I would say, advantage to apply glycolipids into the field. And our strategy also not only to apply glycolipids, but also you will have broths containing other sugars coming from the fermentation of winery residues. So you will have have like a matrix of compounds, they would help and boost the biological process of bioremediation. So you need to think about that. But if you Google about biosurfactants, most of them they have a nice profile of toxicity against some chemical surfactants, and they are sustainable.
Okay, thank you. Neda, do you want to intervene as well, as your presentation was also about surfactants?
Also, as told Ali, it's exactly like this. The biosurfactant has a really low effect on the soil. So also we consider it for the next solution for the remediation technologies. And for our biosurfactants, we are still working on them, and we are studying on them. And till now, we know that they have a really low level of toxicity.
Okay, thank you so much. I want to ask if any other participants want to raise questions, or also my colleagues, the moderators, if you have any questions specific for the speakers. Otherwise, I have a couple, but I want to give you the opportunities. Christian.
Yes, thank you, Natalia. I have questions to Elisabetta. Do you see a possibility to extend this technique to other types of pollutants, for example, to pollutants that are difficult to degrade or persistent, such as, for example, polycyclic aromatic hydrocarbons? I don't know if Elisabetta is always here.
Yes, but I can't make the video. Did you hear me?
Yes,
we
can
hear
you.
Yes.
So sorry, could you repeat your question, please?
Yes, I found your presentation very interesting about hydrocarbon degradation of hydrocarbons. And I wanted to ask you if you see a possibility to extend these techniques to other types of pollutants, for example, to persistent pollutants such as polycyclic aromatic hydrocarbons.
Yes, of course. We have some experience with those pollutants, and absolutely.
We don't hear you very well.
I said that yes. Did you hear me? Yes, we can apply these techniques to other pollutants, such as you said, and we have experience with some of these pollutants, and they work well, these techniques, also with this.
Thank you.
I hope to.
Yes, thank you.
Answer. Thank you to you.
Thank you, Elisabetta, and thank you, Christian. Deji, do you have any question you want to raise?
Yes, I want to raise a question to Neil Bruce. First, I want to congratulate you on publishing on Nature Biotechnology. I mean, it's a very good achievement. I do want to ask. This is a genetically modified plant, right? I mean, I worked in the US for about 10 years, and my experience with local regulators is that it's usually difficult to get approval for genetically modified bacteria when it comes to bioremediation. I wonder, for plant-based phytoremediation, what's your impression? I mean, do you foresee that local regulators would be inclined towards granting approval, or you also see similar difficulties here?
Yeah, that's a really good question, and I'll try and be concise in my answer. So obviously there's lots of examples of transgenic crops being grown across the world, but obviously particularly in the United States. With phytoremediation, it's slightly unusual because there aren't that many examples of plants that have been engineered for this particular application. However, over the years, we've been working very closely with APHIS of the US Department of Agriculture, And it's really a risk-based process. So in order to get deregulation status, we're going to basically need to convince the regulators that the risks are minimal. And there are two key things to be able to show is that one, that the process will not make the plants be more invasive or increase weediness. So do you get horizontal genes transfer to other plants that will enable them to become more invasive, which is one of the things we're doing at the moment, which is very, very unlikely because the genes we're expressing in these plants are specifically for explosives degradation. So they confer no other capability to the plants. So I think that one is going to be, we've got to do the trials and experiments, but I think that'll be relatively straightforward to show. The other aspect is that these plants show we do not make them toxic to organisms that might be herbivores that would feed on these plants. And again, we've done trials in the lab to show that this is not the case. So it's just a case of building up a large body of evidence showing that the risks to the environment are minimal. And then I'm reasonably confident that we'll be able to use these plants on a much larger scale.
Got it. Thank you, Neil.
Thank you very much. I see there was another question on the chat for Dr. Alif, is regarding the yields of RLs using alternative carbon sources and the potential for scaling up your experiment to industrial use. And I think this question is also applicable to all the speakers. So if after Dr. Alif, any other wants to intervene on how to scale up your experiments to a larger scale application, you are invited to do so. So please, Alif.
So I'll reply to this question. I'll address this question to two I would say, parts. The first part is large scale fermentation of biosurfactants. Yes, we succeeded it, and we go for up to 10 liters of biosurfactants now, and we will go for more. This is, and we studied even the degradation of sugars coming from winery residues, and we know exactly what we have inside our broths and cell-free broths. Which kind of metabolites, and we are studying the ecotoxicity of those broths, and now we are aiming, we are writing a proposal to go for 10 meter cubes application of biosurfactants, and hopefully we'll get granted, and we'll go for upper scale using biosurfactants. And this is kind of dual activity, I would say, using biosurfactant and another bio products coming from winery residues, and to see their effects during the bioremediation of contaminated soil. But I agree with you, the field of large scale application of biosurfactants, it's something we need to work on it in the future.
Thank you so much. I would like to hear from Walu and Robert. What is your experience with your proposals, and if you think those can be taken up by industry and can be used for larger areas? So Awalu and Robert, if you are here with us. Yeah,
Okay.
Awalu first.
Hello, everyone.
Yeah.
As for this, my research, it was initially conducted in the laboratory at the laboratory scale. So we hoping to extend it to the field scale, because for us to see the larger impact on this application, we need to apply this technology in real field. So there, after we get the results, then we can conclude on the performance of this application. So that's with regard to my research so far. Thank you.
Thank you. Thank you. And Robert, because I see in your experiment you have closed the circle of remediation.
Yes.
So I would like to hear from you the possibilities for scaling up.
Yes, so basically, actually, the project we are working on, it's also like working on having it in a large scale for industries, because we are looking at using the Miscanthus biomass to actually make paper and make some pulp. So, which means that we want to implement this in large Miscanthus fields, and then also have biochar from the waste from the large fields, and then make biochar. So, and they'll be incorporated back into the fields. So we are still working on the possibility, because we, of course, there are some challenges on how we apply the biochar appropriately in the field for the best results. So we are still working on, but it looks a possibility, since there's already research done on how Miscanthus can be grown like in fields for energy, and how they actually remediate soils. So I think my supervisor, Professor Valentina, will surely add more information to what I have.
Okay.
Thank you so much,
Do
Robert.
you hear me?
Yes, Valentina,
Do you hear me?
Yes.
Yes. Congratulations with your good event, Natalia, by the way, and all other participants of the event. I want to just briefly add that we are doing field experiment within this project, Cornet, and we have the second year field experiment when we incorporated biochar, different doses of biochar and different type of biochar to the field in Chomutov in the Czech Republic, and we are monitoring the quality of biomass, and simultaneously we are monitoring the change in the soil microbial communities and generally changing of parameters of soil. So we will report, I guess, maybe next year, because it would be third year of experiment, and for Miscanthus it's good to have at least 3 years of experiment. So we are doing not only lab experiment, but we are doing also field experiment, and this field in Chomutov is post-mining field, which is slightly contaminated but by different trace elements. This is what I want to add for implementation. And this Cornet project is combination of phytoremediation, actually phytotechnology with biomass production, and processing of biomass to different bioproducts. This is German parts of the project. So I would be happy, and my group would be happy to present next year the results of the project. Thank you for giving me the floor. Thank you. And thanks for very interesting event. Thanks.
Thank you, Valentina. Well, we have reached 11 o'clock this morning. It has been a really interesting session. I want to see if my colleagues, Deji or Christian, have any other final question. No?
No.
Deji? No. Okay. So I think we have reached the end of this super nice event, and I would like to give the floor to my colleague Christian to give some final words and to close this event. Thank you again to all the presenters. It has been really interesting session. Thanks. Christian,
Thank
please.
you, Natalia. Yes, on my side, I would like to thank all the speakers for the presentation, which gave a very nice idea of different kind of bioremediation and phytoremediation project. And I would like to thank also all the participants for attending these sessions and for the nice discussion in the chat and at the end. And I would like to remember the next session will begin in approximately 30 minutes, and the topics will continue on oil and hydrocarbons impacted sites at 11:30. Thank to all, and I hope we have the opportunity to see us online or to hear us during this very interesting week at Remtech Europe. Thank you.
Thank you again to all. And yeah, you have the event planning on the screen. So don't miss the upcoming sessions that are also very interesting for today. Thank you, everyone.
Thank you. Bye-bye.
Bye.